So my lab actually, we don't work on ribosome. We work on one of the very interesting problem of the biology. And so, as you know, eukaryote cells, they have nucleus and they have cytoplasm, right? And there is a constant communication between nucleus and cytoplasm, and it happens through channels which are present on the nuclear envelope. Nuclear envelope is a very closed, like lipid bilayer around the genetic material, and that separate the it and like from the cytoplasm. So if something has to, the message has to be communicated to the genetic material, it has to go through this nuclear envelope. And on this nuclear envelope, there is a group of about 30 to 34 proteins which are present in a complex, and that complex is known as a nuclear pore complex.
Hey, hi, welcome to Biotech Talks. We have discussed about antimicrobial resistance in the previous episodes of Biotech Talks, right? And it is a well-known fact that we need new antibiotics. So the question to ask is how to develop new antibiotics, and one of the answer is through structural biology. So you use various methods in structural biology like electron microscopy, X-ray crystallography, NMR spectroscopy to determine the structure of ribosome of a bacteria which is showing antimicrobial resistance. Once you determine the structure, you have to make a molecule that will inhibit the activity of that ribosome. And once the activity of the ribosome is inhibited, the bacteria will not be able to produce proteins and its growth will stop. And that is how you deal with the problem. So what is structural biology? What are the different methods in structural biology? And much more will be explained in this episode of Biotech Talks. So today with us we have Dr. Radha Chauhan. She is a scientist at NCCS. In her lab, she is studying the nuclear pore complexes of eukaryotic cells. So fascinating research and a wonderful person to explain it. I hope you enjoy today's episode of Biotech Talks. Hello ma'am, how are you?
Good, thank you. How are you?
So starting with the first question of the podcast, what is the importance of 3D structures in life science research?
So 3D structures are really very important. It's like visualizing something. So if you look at any object and if you don't know how it looks, you cannot understand the object. So 3D structure in biology are very important, because that's how we the how small is cell, right? Cell is really small, and inside the cell there are more than 30,000 proteins, and they are even smaller. So once we get to know the structure of these proteins, we get to know the function of these proteins, and that function lead us to better understanding how cell works, how they communicate with each other, or how they are important for public health. So altogether, if you have first step like the structure of these protein or any biological macromolecule, you can actually understand the processes of biology. So they are very important, and one important milestone in understanding the complex biological functions like mitosis, transcription, replication, you just name it. So they are very important.
So, to determine the by determining the structure of proteins, we can determine its function.
Yes, yes, yeah, yeah, yeah.
So, what are the actual techniques to determine the structures?
So, as we know, biology is a very new science, like unlike the many other science. It's like, because as the technology keep developing, we start getting better understanding of the biology. In structural biology area, techniques and technology is a very important aspect, and there are many methods which can be used for the determination of these structures, and they were developed or being getting developed over the decades. So I think one of the foremost and important method is X-ray crystallography, where what we do, we purify the protein and we make these protein into crystals, like the way you guys or anyone can grow like sugar crystals or calcium chloride crystals, right, or copper sulfate crystals in our high school project. Same way you can actually take these purified protein and you can grow them into protein crystals, and these protein crystals then we freeze in like a minus 100K temperature, and that crystal then we expose to X-ray source. X-ray is one of the aspect of the light source, one of the wavelength 1.5 angstrom close to that, and that wavelength we use to expose these crystals and collect the diffraction data. So diffraction data is like, if you crystal, is the systematic arrangement of lattices, and when you expose through X-ray you get the diffraction data, and that we process and we can get the 3D structure of the protein in angstrom resolution, like 2 angstrom, even better than 1 angstrom, where you can even see hydrogen atom. So that's how we generate. So what we do, we actually build the models. So we know the protein sequence, we you can look at the electron density map, which is derived from these diffraction patterns, and you then fit looking at the density map, alanine at alanine position, serine at serine position, and you can build the model, and that's how we build the structure. So that's one method, X-ray crystallography, and which is almost 100-year old, and a lot of development has happened, and the method is still getting advanced in coming years. The second foremost important method is now emerging as an important method. It was not that important a few decades back, but since 2012 a lot of development happened, and electron microscopy has become a very powerful method. So in electron microscopy, instead of diffraction pattern, what we do, we collect images of transmitted electrons, and lots of images, lots of data we collect, and the protein particles are distributed in random manner, and then we we average them together, and we get a better signal to noise, and we get the angstrom level resolution from this. So this method is something which just getting developed now, and a lot of major technological advances were done since 2012, and now method is accessible to everyone and can be used. The other methods of same caliber of angstrom resolution level is the NMR, nuclear magnetic resonance, and so that also can be used. That's where we collect the data of the nuclear magnetic resonance, and it's a very complicated method, and we cannot use this method for bigger than 30 kDa protein, but it's one of the very important method, because in solution you get data, and in solution you can understand the dynamics of the protein. So these are the three major method, and after that there are other supporting method where you can actually understand some structures or at the angstrom level resolution, instead of fluorescence microscopy and spectroscopy method, FTIR, CD, which tells you at the secondary structure level.
Okay. So, in our conversation before this podcast, while descending the questions, you were explaining about the importance of ribosomes
Yeah.
in determining new antibiotics, in finding new antibiotics, and that was very fascinating. Can you please elaborate it?
Yes, so ribosome is a very important molecule in our cell, and it's the major workhorse for the protein synthesis. So all protein synthesis happens through ribosomes, and when so ribosome is a very big molecule. It's actually a complex of protein and RNA, and it's about 2 to 3 megadalton. In human it's close to 3 megadalton complex, and so this structure is determined by both X-ray crystallography and electron microscopy. And we know before, prior to this, that many antibiotics which were discovered in 1960s or like in 1950s, like tetracycline, which they were targeting this protein synthesis, and they were binding to ribosome. But the mechanism part, as I said, that the visualization is very important tool we have. But the mechanism was not known. So once we started getting the structure of ribosome, we started getting the structure of some structures in complex with these classical antibiotics. So now we know that how one antibiotic is bound to the ribosome in the pocket. And what you can do now, we have lot of computational tools, and even coupled with experimental tool, what you can do, you can actually now know how to change this classical and one antibiotic into now a derived antibiotics. For example, it's a tetracycline. you can actually now change the chemical structure of tetracycline and figure out that how you can actually improve the binding or how can we generate the new. And of course, coupled with the experimental science, you can come up with a new series of antibiotic. So that's why the ribosome structures were very fascinating. When we started getting ribosome structure, they were very exciting, and they were a new hope that eventually we will have a new set of antibiotic, because MDR or multi-drug resistance is one of the biggest problem we are facing, and in fact in coming years it will become one classical like a major challenge to the human health. And so having these structures of the targets where old antibiotics were binding, we can understand better mechanism if we have those structure, or we can actually design new classes of antibiotics or any new drug molecules, and that can be help us, and that will empower us to tackle the multi-drug resistant challenges what we are anticipating in coming years.
So yeah, if a bacteria is resistant to particular drug, then we just need to, like we have the
Yes.
ribosome structure, we just need to develop a new
Yes.
antibiotic which can fit. I mean, it's
Yes.
easy to
Yes.
say, but it's
Yes, yes, it
this
is.
is the
It is
hope we
doable.
have.
It is doable. Only
Yeah.
thing, it will take years, and you need certain infrastructure and certain expertise. But it is certainly doable, and that's where the world is going in this direction right now.
Okay.
And there are many researchers across the globe who are working in this direction, and they are trying to come up with the new drugs and better drugs, right? Whatever we have, we know there are certain drugs with the side effect, or they don't work effectively on the entire population. So this is how we are improving our drugs and molecules.
Is this field playing any role in personalized medicine?
Eventually, yes, it will. So once we start understanding the all nitty gritty differences at the structural level of, for example, ribosomes in your body, in my body, right? And so all the nitty gritty, like, so once it's like, so in biology we work in very systematic way, right? We first get the basic. So in fact, first structure was determined was of the prokaryotes. The first ribosome structure came from the prokaryotes, and now actually few years back the human ribosomes, yeast ribosome structure started coming, right? So it takes us time to get all these structures. It's not that straightforward, but it is like, this is how we do research. And now as of today, we have now like many pathogens ribosome structures. We have many ribosome type from the mitochondrial ribosome type and the human other like variants of ribosome. And now you can actually understand both structure, and you can understand the antibiotics how they were binding earlier, and now you can come up with the new. So once we start understanding individual variants, like, of course it can do, but it will take time. It's not something gonna happen in next 5 years, but this is where the research is going in this direction.
So what research is going on in your lab?
So my lab, actually, we don't work on ribosome. We work on one of the very interesting problem of the biology. And so, as you know, eukaryote cells, they have nucleus and they have cytoplasm, right? And there is a constant communication between nucleus and cytoplasm, and it happens through channels which are present on the nuclear envelope. Nuclear envelope is a very closed like lipid bilayer around the genetic material, and that separate it and like from the cytoplasm. So if something has to, the message has to be communicated to the genetic material, it has to go through this nuclear envelope. And on this nuclear envelope, there is a group of about 30 to 34 proteins which are present in a complex, and that complex is known as a nuclear pore complex. And that is the only channel between the cytoplasm and nucleus, and all transport happens through it. So now if you imagine, this is like one of the gate or the toll gate, you can see that's where, like in the road, suddenly this toll gate comes, and that's where one. But on the road we are doing only one way of things, right? We are going or coming and paying toll for that. But if you imagine nucleus and cytoplasm, there is a thousands of events happening simultaneously, right? There is a protein synthesis happening in the cytoplasm, and for that protein synthesis, mRNA has to go out of the nucleus, right? And it will go through nuclear pore complex, and it will attach to the ribosome, and protein synthesis will start happening. Suppose cell receive a signal at the plasma membrane, right? And that signaling molecule trigger a event, and that trigger, like a factor, has to go inside the nucleus, transcription factor. It has to go. So the nuclear pore complex has to sense that, okay, it has to go inside the nucleus, and it will allow to go inside the nucleus. And this is how we control the cell, control the gene regulation. So imagine if this gate is not there, actually everything will collapse, right?
Yeah.
But this being having a gate there, it actually control your uh, all kind of event, gene regulation, and mitosis. So at the time of mitosis, cell divide, that also is tightly regulated, but nucleopore complex do play a role. That genetic material is encapsulated immediately after that. Any kind of like variation happens, like a cell has to go into autophagy or what, all those related phenomena are controlled through a nucleopore complex. So what we are trying to do, we are structural biology lab, and we use the tools of structure determination. In this case, in our case, we are using X-ray crystallography and cryo-electron microscopy to get the high resolution structure of the NPC. It's a 34 protein complex. It's about 120 megadalton. So ribosome was only 4.5 or 3 megadalton complex we are talking about. So you can imagine 100-fold like bigger than this. And so we are trying to determine the structure of some of these components. Out of 34, we are working about 10 to 12 nucleopore in our lab. So that all these proteins are known as nucleoporins, and so they exist in complex. And what we are trying to understand that how they are playing their role in forming this nucleopore complex. So what we do, we biochemically purify these nucleoporins, and we are working on the human nucleopore complex, which are very, very important for the human health. There are many of these nucleoporins are linked with chronic kidney disorders, and they are linked with the neurological diseases, IBS, and infantile bilateral striatal necrosis. So it's a genetic disorder in which the brain of the infants, they start getting into necrosis. And where one of the nucleoporin we are working on is very important for cardiac health. So at like arrhythmia, it causes arrhythmia. So like, but nobody knows the mechanism that how these proteins are important for these functions. Again, that because we really don't understand the mechanism and structures. So what we are doing, we are actually asking very fundamental question, that how these nooks are important for the function. And once we get to understand their important role in NPC assembly, then we can understand its function. So we purify these nucleoporin. We try to form a complex by biochemical reconstitution, and once we have, which my student takes several years to actually reconstitute these complexes. And once we have that ready, then we take it to either, if it is a suitable project for X-ray crystallography, we actually start forming protein crystals out of these proteins, or we actually start optimizing for sample for cryo-EM method. And then we get the structures, and of the resolution, we really aim to get the 2 to 3 angstrom resolution, where you can see each amino acid. And once we have these structures, then it's very important. One of the recent study in our lab on one of the NUC 155, as I was mentioning, it's important for cardiac arrest. So what happened, there is a one genetic mutation present on NUC 155, and that causes the atrial fibrillation. And this disease in infants, when you have these homozygous mutation, infants actually dies because of that, so they don't survive anymore. But the mechanism was not known. So from our structure, which we determined through cryo-EM, at the biochemical level, we could explain that what was happening at the protein level. So what was happening, that in this disease mutation, this nuke is very important to interact with other neighboring partner, and this mutation is happening where it is in in a vicinity of other interacting protein sites, and that was getting affected, and the interactions mapping is getting affected, and that's how the function is getting, and like dominant of like in this kind of event. So it's an ongoing research. We are still trying to understand that how this nuke is important for more for cardiac cells rather than for everywhere else in the body. But this is how we build hypothesis. So once we have a structure, once we understand the independent biological phenomena, we try to really now come up with a hypothesis that how this disease is happening or leading to this disease, and then once you understand how disease is happening, then you can actually think of having interventions, how you can actually control it, how can you can prevent it.
Yeah.
So that's how the we goes in a step by step manner in this research.
So after determining the structures, we get the mechanism, and on the basis
Yeah.
of that, we can develop therapeutics.
Yes, yeah.
Can you explain some more therapeutics developed on the basis of the structure, determining the structure?
So actually there are a lot. So nowadays, so there is a term structure guided drug design, and so a lot of big pharmaceutical companies like Pfizer, Novartis, and so what they have a very strong and a very big drug discovery programs, and which is really driven by structures. So in this structure-based drug design, you have a structure of important drug target. For example, in cancer, if you know, in the breast cancer, like the BRCA protein, it is a very important, not only as a marker but as a drug target, right? So once we start understanding the structure, and you know that okay, how to really break this pathway, like how BRCA is important, so you can break this pathway somewhere. So that's where and chemist so drug discovery is a very interdisciplinary research area. It's not like one lab can do it, like, so that's why pharmaceutical company are more equipped to do that. So you have a medicinal chemist there who has a libraries of synthetic compounds, right? And computationally, either you computationally screen which compound will bind and can inhibit the function or induce the desired function, whatever function you want to look at, or you can do experimentally also, that you take one your cell based assay and screen these compounds, and you will get certain class of molecules or group of molecules. And then having this structure and having the structure of your compound, which we call lead molecule, you can actually optimize, and you can fine tune this, and then eventually it will come up as a potential drug molecule. It's a long process. I mean, it's the drug discovery process is 5 to 10 years depending on which phase you are, and but you can improve. This is one example. Other example, because it's a pandemic time, one can think of SARS-CoV-2. For example, in SARS-CoV-2, everybody knows that spike protein, right? Everyone knows spike protein. So spike protein. So the reason is that spike protein is important because it binds to ACE2 receptor in our body and helps the virus to amplify. And if you block this binding, you actually stop virus multiplication in our body, and you stop the infection. So what our field has contributed, or structural biology field, so there is like at the global level, people really chase this complex and try to determine the complex of spike with ACE2, and try to understand in not only of wild type virus but all kind of variant what we have seen, including Omicron. This is in real time. In last 2 years we could see that how the Wuhan strain spike protein was binding to the ACE2, and how our body will generate immune response, and in immune response antibody, and which can neutralize this virus, they were binding. Okay, and then now the latest one is Omicron spike protein, and we have structure of that, 3D structure of that at high resolution, and how it is binding to ACE2, and how other neutralizing antibody they are binding, and that helps in understanding the vaccine development also. So now we understand that why like Omicron had so many breakthrough infections, and why like what kind of changes we really need to do in the drug discovery or program, including antibodies, like which can lead to that kind of like a widespread usage of the new treatment. So it's very important. So this is one of the example where we have seen in real time how the structures of the pathogens and host receptor helps in understanding the disease, and how and the impact is tremendous, because that's how we really realize that how doctors can think of new treatment, new guidelines, and all came based on that.
So, the conclusion that I made, that earlier, whatever the drug discovery and whatever we were doing, it was just trial and error. Like discovering, like if it works, it works. But because of this 3D structures, determination of 3D structure, we can be more specific.
Yes, initial I wouldn't say that it was trial and error, but so drug discovery can have two independent phases. Like once, so either you solve the structure. Imagine if antibiotics were not discovered earlier, like in
Yeah.
1950s, 60s, right? Ribosome structure came in 2000, like 1999 onwards, like high resolution structure. So 50 years we would not, if we keep waiting for structure
Yeah.
and that. So it's a both like parallel pathway which works in biology. So everybody is not structural biology, which is a good thing because there were a lot of like scientists who at the cell level try to understand how to stop. So like they didn't have the structural information, but inside the cell they could actually see that tetracycline or penicillin works on different pathway, but like how it was blocking the bacterial growth, right? But once we had the structure of the drug target, you can improve very fast. So it's a combination of both. Hit and trial is really needed, but once you have the structure, that can really impact the speed of the new discovery. And so if, for example, SARS-CoV-2. So SARS-CoV-2 is from the family of coronavirus,
Yeah.
right? We had earlier MERS and other diseases, right, from the same family, and we had some structural information available from there. But once we had this brand new virus circulating in the population, and once we know the sequence and we know that this is a spike protein, we can actually understood better when we had started getting the SARS-CoV-2 spike protein structures, right? So otherwise it's a random trials, and so this speed up, this is the result that in SARS-CoV-2 case, within 2 years we had. Now actually in coming year you will see a lot more promising treatment, new drugs, and a lot of new things are coming because of this faster structural availability. So that does help and speed up. But the new drug discovery program is time consuming. It takes time. For example, cancer, again, we have a lot of advantage of having the structures of drug targets there, but it's a complex disease and you need to understand what is the side effect, what is the, how it is overcoming that drug, like the cell system. So you have to keep improving and keep working on that. So that process takes time. Yeah.
Thank you, ma'am, for your things.
Yeah, thank you very much. Yes.